Welcome back, ladies and gentlemen. Happy Thursday. Please sign in with a P for present in the stream if you have not already. Today we're going to talk about how we know exactly what parts of our DNA need to be coded and transcribed and translated into a protein. And then we're going to get some more practice because tomorrow you will be having a quiz. So everyone, make sure your cameras are on. Roy, I know you're going to be leaving at some point for a doctor's appointment. No problem, kiddo. I get it. Thanks for letting me know. Okay, so thanks, Jasmine. Who could tell me what the 3 steps of protein synthesis are? Real fast. Yell it out.
Transcription, translation, and elongation.
Elongation, right? We're making a long chain of amino acids that makes a protein, right? Excellent. Okay. So today we're going to look at how we actually go from DNA to messenger RNA, right? We need an enzyme. There's an enzyme there that helps us read DNA. And so I'm going to go ahead and share. This is a 1-page of notes, and we're going to look at a video, and then we're going to do some more practice. So I've taught you a skill, right? I taught you the language difference between DNA and RNA. And there has to be a way to translate from DNA language to RNA language, and the enzyme RNA polymerase helps do that. So write down in your notes, RNAp is RNA polymerase. And as you guys know, anything that ends in ASE is a protein enzyme. And so RNA polymerase is responsible for figuring out what parts of DNA actually do we want to translate. Because some segments of our DNA code for traits and others don't. It's just extra DNA. And so before we begin with this, let's take a look at this picture. Here we're in the nucleus and we have a DNA molecule. Long strand of DNA. And as you can see, there are 1, 2, 3 genes along this strand. Now, when we say we have a gene, what's a gene? A gene is a series of DNA bases that code for a trait. And so some parts of DNA make up a gene, while other parts of DNA do not. You might say it's extra. You know, some scientists, some researchers actually called it junk DNA, not garbage DNA. And there's a difference. And I know the subtlety in the language you have to catch on to. Junk, right? Junk is something that maybe you don't need, but somebody else might want. Like if you go to the junkyard and you're somebody who likes to build stuff with their hands and you need some materials, you can go to the junkyard and grab some materials and build something that you would need. So it's not garbage. You just don't know what you can use it for yet. And that's what some parts of our DNA are like. Does that make sense? Give me thumbs. Okay. So when we go ahead and we look at these parts of our DNA, we see that there are segments that will be read by RNA polymerase and used to make messenger RNA. Those parts of our DNA are called exons. Please write that down. So RNA polymerase will read exons, and then there are some parts of our DNA that do not code for anything. Those are called introns. And so the whole job of RNA polymerase cuts the fat from our DNA, right? It's like you have a nice piece of steak. You remove the fat to enjoy the rest. And so really all that you're using then is a streamlined version of all the important parts that will help you build a protein. Does that make sense to everybody? Thumbs? So we're trying to streamline the process of going from DNA to RNA to figure out, well, how do we make a protein as quickly as possible? Let's get rid of the pieces that don't really mean anything right now, and we'll focus on the pieces that do. So the exons are important. Doesn't mean the introns are not. Eventually, through evolution or other means, those introns may have an important part in our cellular development. At the current time, they don't. And so this streamlining process becomes very helpful when we need to make proteins very quickly. So let's take a look at a video that pulls all of this together. Right? Matteo said that we have transcription, then we have translation, then we have elongation. Let's look and see what this looks like in a video. I'm going to go ahead and mute myself so it doesn't echo.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In human, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
How is that? Does anybody have questions? So show me thumbs if that was making sense to you. Okay. Good, good, good. You guys ready for another practice problem? Going from DNA to protein? All right, let's giddy up. Here we go. Put this in your notes. I'm going to go ahead and share with you. I'm going to flip over to the dry erase. I think it would help. So remember, we start with DNA. So we're going to go ahead and use RNA polymerase to read our DNA and just use the important parts to make a strand of messenger RNA. Each little 3-base segment of messenger RNA is called a codon. Then we're going to go ahead and make sure we have a complementary anticodon. So you're going to have a row of anticodons that you write out. And then finally, you're going to have to tell me what amino acid each anticodon brings by using your chart. And this is the tricky part, right? We use our chart with codons, not anticodons. So you'll have a red line, an orange line, and then in green, or this last one here, will be the amino acids that are getting linked together based on what you read. It's 10:24. I'm going to give you guys until 10. I'll give you 4 minutes. 10:28. Write this in your notes, get your answers out, and be ready to answer, because I'm going to call on people to give me codons, anticodons, protein, or amino acids. So be ready to answer, please. Francisco, are you wearing glasses today? Show me a little bit more of your face here. I'm almost. There you go. There it is. There it is. This is really all that I see for some people right here. It's just I'm gonna draw Kaylee next. That would be easy, right? There's Kaylee. Is that true? Wait. Hold on a second. Gotta get the old ceiling fan in. Right? Is there a light bulb there? Light bulb? Is there a pull chain? I'm not sure. It's a light switch. Sorry. All right, let's take a look here. Let's take a look. Okay. Now, we are going from DNA with the help of DNA polymerase. Excuse me, not DNA polymerase, RNA polymerase. Right? With the help of RNA polymerase, we're going from here to here. So what is the codon for this triplet? Sameda.
AUG.
Money in the bank. Beautiful. Colton, what's the codon for TCA?
AGU.
AGU. Excellent. What is the codon for AGG, Roy?
UCC.
Great. Goon Squad. Jess, what's the codon for TCC?
AGG.
Perfect. Jasmine, what's the codon for ATT?
UAA.
Done. So now that we have our codons, it's time to find our anticodons. Michael K., what's the anticodon to AUG?
UAC.
Beautiful. Kylie, what's the anticodon to AGU?
UCA.
UCA. Reese, what's the anticodon to UCC?
AGG.
Yep. Daza. Jacob, what's the anticodon to AGG? Beautiful. Marta, my dear Marta, what is the anticodon to UAA?
AUU.
AUU. Perfect. Each one of these anticodons is bringing an amino acid. Now, did you guys notice that we actually started with the start codon? So our life was really easy for this one, wasn't it? We got pure exon here, didn't we? So if this is our start codon, we take a look at our chart. AUG, A, somewhere in this big row, U, now we break it down into here, and then G is the third letter. Whoop, right here. Methionine. Methionine. So I'll just put meth. Victor Garcia, what is the amino acid that is brought next?
Let me just the chart's loading. Let me just put it up.
Remember, we're using AGU, not UCA. Use your codon. Don't use the anticodon, or it will jack up your work.
All right. It's serine.
That's it. Serine. Excellent. Rose petal. What's the next amino acid, dear?
It's also serine.
Yes. Yes, it is. And then finally, Anna, what is AGG? What's that last amino acid?
It's a stop. Oh wait, no, sorry, hold on. Arginine.
There we go.
Sorry.
No, you're fine. You caught yourself. Great job. So arginine, arginine. And then everyone take a look at UAA on your chart. You will see that it is, in fact, a termination factor, otherwise known as a stop codon. Yes? Raise your hand or your thumb if you feel like you're getting this, and this is pretty straightforward. Love it. Love it. You guys are crushing it. First period or second period honors, they are struggling, man. They are on the struggle bus. They're on the Twinkie bus. We should send them back to Elm. Right, Kylie? You know what I'm saying? Really? Let's clean it up here at the high school. It's only for the strong. Let's just send them to Ridgewood. Let them take care of it. Bogus, man. So practice, practice, practice. Tomorrow you're going to have a quiz. You're going to have a question just like this. You're going to have to know. I'm going to give you a chart so you don't have to have a chart ready. You'll have a chart ready to roll in the quiz. Go through your notes. Make sure you know what the central dogma of biology is. Make sure that you understand the language. What's a codon? What's an anticodon? All that good stuff, right? Exons, introns. So you're going to have a quiz tomorrow in class. I will give you a practice problem tonight. I will also send a key for that practice problem. Sound good? Just make sure you guys try it first before you look at the key. And if you have questions, as always, please reach out. Any questions to close it up today? We good? All right, gang. Have a great day. See you tomorrow.